Greetings, humans. I'm going to try to explain cell division in a way to hopefully reteach it, so that will help prepare you for the test on Monday. So we've been talking about why cells divide. The two main reasons that cells would divide are going to be to repair anything that's damaged. So like when you cut your skin, you need to repair those skin cells. Your skin cells are actually always dividing, so repair anything. And also when you're growing. So like my kids are continuously growing, so their cells are continuously dividing for that growth. When we've been talking about cell division, cells exist in something called a cycle. So we made cell cycle spinners to kind of visualize this cycle that cells are going to go through. And it's a cycle because it's going to repeat. Okay? The majority of this cycle, I guess I should have done it in a different color, but the majority of this cycle is called interphase. Interphase is just when the cell's being normal. It's going to go through normal cell growth. It's going to go through normal processes. It's going to obtain nutrients. It's going to go through respiration. It's just being a normal cell. In G1 and G2, oops, that's a 2, they're going to go through growing. So it might copy the organelles. It's going to make more stuff and things. It's preparing to make two cells from one. In the S phase, though, it's important to know because that is when DNA is copied. If you are trying to do honors, you would have learned about DNA replication through that Nearpod posted on Schoology. The S phase is where DNA is copied. We want to copy the DNA because we're going to go from one cell. That cell is going to divide into two cells. So whatever DNA is in the original cell, we need all that DNA to be in those daughter cells because the parent cell is going to be identical to the daughter cells. They're going to be exactly the same. They're going to have exactly the same DNA. All the parts are going to be the same. They're identical. I should write that. Identical daughter cells. That is important to know. So like I said, interphase is what the cells are going to spend the majority of their time in. After interphase, after the cell is prepared to divide, it's going to go through two parts. One is mitosis, and in mitosis, we're going to divide the nucleus. And a great, easy way to remember mitosis is to think PMAT. That will help you remember the phases in mitosis. And then after mitosis is going to be our final phase, our final part of the cell cycle, which is cytokinesis. And that is where you're going to get the division. Divides nucleus. That's where you're going to get the division of the nucleus. The rest of the cell is going to split apart. So this is the overall process of cell division. And then now I'm going to go through each individual phase. Colors bled through a little bit. So this is the cell cycle from the point of view of the actual cells. So at the very beginning, where the cells spend the majority of their time, is going to be an interphase. Interphase is not going to look like much. We, of course, are going to have a nucleus, which try to make different colors. And inside the nucleus, we are going to have chromatin. What color? We'll make the DNA purple. The chromatin is going to be those chromosomes uncoiled. The DNA is most looks like chromatin most of the time. We're also going to have these other structures over here. And they got copied during interphase, called centrioles. And the centrioles are what's going to be producing the spindle fibers later. So interphase is where we're going to have G1, S, where DNA is copied in the S phase, and G2. And the cell's going to spend most of its time into interphase. When it's ready, it's going to enter mitosis, where the nucleus is going to divide. There are 4 phases in mitosis that you can remember as PMAT. P, the first phase, is called prophase. And now we need to divide the cell, so we are going to first make those chromosomes condense into, that chromatin condense into chromosomes so they can actually be easily organized in the cell. And I made the DNA purple, so I'm going to draw my chromosomes purple. They exist as this X shape. They exist as this X shape because they're duplicated. So when we're looking at that chromosome, when it's that X shape, that is a duplicated chromosome. Each side of that chromosome is equal to each other. So this side is equal to that side. It's duplicated. We want to duplicate the chromosome so we can divide it, and each cell is going to have all the DNA. In prophase, we have a couple things happening. We saw that the chromosomes condense so we can actually see them, and they're going to be much more easier to organize. The nuclear envelope, which is going to be the outside structure of the nucleus, is going to disappear. It's going to dissolve. And then these centrioles are going to start producing these spindle fibers. So those spindle fibers form. You're only going to see the spindle fibers and the chromosomes during mitosis because that's when we need them. Right, so after prophase, now that we have chromosomes and we're getting rid of the nucleus, now we need to start organizing those chromosomes in a process called, or a phase called metaphase. And in metaphase, the cell is going to line up those chromosomes in the middle. What is going to light up the chromosomes, or help this process, are these spindle fibers. The spindle fibers are attached to this centriole. I don't think I labeled spindle fibers yet. They're going to attach to that centriole on the end. And then on the chromosome, it's going to attach to the centromere. The centromere is the center part of this duplicated chromosome that's going to hold the two chromatids together. So in metaphase, the chromosomes line up, and spindle fibers attach to centromere. Okay. All right, and then after metaphase, now that we have organized the chromosomes, those chromosomes are going to be pulled apart by the spindle fibers. And so it kind of looks like they're being dragged away because they're physically pulling them apart. So that's why they're kind of shaped this way, because before they're shaped the other way, and they're being pulled apart from each other. So the sister chromatids are separated by spindle fibers. Then we're done dividing the chromosomes, so we now need to get them together by creating 2 nuclei. So 2 nuclei form. So these nuclei are going to reform. My nucleus was red in prophase, so I'll make it red again. So this is a process. So at the beginning, there's going to be no nuclei, and then at the end, it's going to be a complete nucleus on each end. The chromosomes are going to uncoil, and then the spindle fibers disappear. Okay. And I'm just going to draw them a little bit longer than they already were, because they're slowly uncoiling. They're not completely uncoiled. Okay? That is the end of mitosis. And then after mitosis, we need to complete that division into 2 cells. So that cell is going to start pinching. We have complete chromatin in both cells. The nuclei are complete. This process is called cytokinesis. And right here, that pinching is technically called cleavage furrow. So pinches into 2 daughter cells. Okay. And now that cell process will be complete with 2 cells that are completely identical to that original cell. Email me if you have any questions, and hopefully you'll be ready for that test on Monday.